Our next speaker will, I think, give us some examples of the application of precision medicine. So I welcome Professor Chris Semsarian, who's a cardiologist and over the last 20 years has developed genetic cardiology at the Royal Prince Alfred Hospital through his research at the Centenary Institute. His focus is on the investigation and prevention of sudden cardiac death in the young, particularly amongst children and young adults. And he is the co-lead of the cardiovascular genetic diseases flagship of the Australian Genomics program. And we'll hear more about that from Kathy North during our panel discussions this afternoon. Chris has also led major community programs in the area of prevention of sudden death, including defibrillators, which we now see in all public places, as well as organised programs to teach CPR to members of the community. And he'll now present to us, I think, some very practical examples of the application of precision medicine.
Thank you very much, Warwick, and it's a real privilege and an honour to be asked to be part of this program, and I thank the programming committee for that opportunity. So what I'd like to do in the next 15 minutes with Marcel's backdrop is to give you some very concrete and clinical examples of how we're using precision medicine today in cardiology practice. I think there's four things that have occurred amongst many which are really important in the field. Firstly, the sequencing of the human genome, which it felt like yesterday that Bill Clinton said this is the book of life and was on the news, and 20 years since that moment, so it's been amazing. Secondly, Barack Obama, the President of the United States, in State of the Union address in January 2015, when he coined the term precision medicine and talked about delivering the right treatment in the right patient at the right time. And then we have the amazing advances in technologies from Sanger sequencing not many years ago, now being able to do panels, exomes, and genomes relatively simply. And as alluded to in the last talk, the cost of the sequencing of the human genome has come down dramatically over the years. So all these particular changes have been really important in setting the platform to be able to do precision medicine. So I'm going to give you a two-slide primer of inherited cardiovascular diseases. There are at least 40 different cardiovascular conditions which are directly caused by mutations in genes that we're born with. And no part of the heart is spared. So whether you're a cardiomyopathy person, a muscle person, an arrhythmia person, vascular abnormalities, metabolic diseases like familial hypercholesterolemia or congenital conditions, all parts of the heart are affected by genetic mutations leading to disease. And the second feature of these conditions, there are some common features. They're often autosomal dominantly inherited. So if I've got the condition, my children have a 1 in 2 chance of having the same problem. Generally younger population, crazy sort of clinical diversity where within the one family people live to 100 with no symptoms and then others die on a football field at the age of 20. They're genetically heterogeneous because there's many disease genes involved in these conditions, and a specialised multidisciplinary clinical approach is a very important way to manage these families. So what I want to do now is give you 5 very simple examples of how we're trying to start to use precision medicine in our clinics. The first is in precise genetic diagnosis of cardiac disease. So what's happened over the last 20 years is that all of these diseases have been redefined because of their genetic basis. So for example, hypertrophic cardiomyopathy 60 years ago was called a tumour of the heart, a cancer of the heart, whereas we now know that it's a disease of the sarcomere. The arrhythmia syndromes like long QT syndrome are now called ion channelopathies because all the genetic causes encode either sodium, potassium, or calcium-related genes. And the list goes on and on. Why is this important? It's important because it helps us define our diseases. So let's look at one of these as an example, hypertrophic cardiomyopathy, fairly common at 1 in 500, clinically heterogeneous disease. Hypertrophy is the feature, and what I see when I see the patient is the image of the echo you can see up the top right-hand corner with a thickened myocardium, and that tells me that there's hypertrophy, there's an underlying cardiomyopathy, and I call it hypertrophic cardiomyopathy. And we know through genetic studies over the years that these cardiomyopathies, hypertrophic cardiomyopathy, are caused, as I said before, by mutations in genes which encode the sarcomere proteins. But what genetics has allowed us to do is through expanded genetic testing, identified these hypertrophic cardiomyopathy phenocopies. So when I see the patient, I see the echo, I see the thickening of the heart, and I call it hypertrophy, but we now know that it's not all hypertrophic cardiomyopathy. So it might be one of these mimickers, a glycogen storage disease, Fabry disease, Danon disease, amyloidosis, etc. And why is this important? It's important because we're completely different way of treating the patient. If it's hypertrophic cardiomyopathy, it might be beta blockers. But if it's Fabry disease, we replace the enzyme. If it's Danon disease, we know that in this X-linked disorder, all males by the age of 20 have died suddenly or have had heart failure or have required transplantation. So we accelerate our therapeutic approaches. So knowing the basis of the hypertrophy is very important, and this may account for at least 10% of the patients we see on echo that have hypertrophy. The second is the use of precise genetic diagnosis in families. The top right-hand corner just reminds me, this is the image I use to teach my clinical cardiology colleagues what autosomal dominant inheritance is. The father has horizontal stripes, the mother has vertical stripes, and the child has hatched shirt. And what we know in these conditions is that if you just have the clinical diagnosis, all first-degree relatives at some stage will require ongoing lifelong clinical screening to see if they do develop the condition or not. But as soon as you find a genetic cause in the proband, you can then test other family members like cascade testing to more precisely define who is at risk and who is not at risk. And the important clinical implication here is that the ones that don't carry the mutation can be released from lifelong clinical surveillance, while those with the mutation can be monitored a bit more closely. Thirdly, in the area of precise genotype in guiding therapy, I've mentioned that already in cardiomyopathies, but I want to share with you the story of long QT syndrome. We know that long QT syndrome usually is acquired due to drugs that we give patients that cause long QT prolongation. But what I'm talking about here is genetic long QT syndrome. Prevalence a bit less common than the cardiomyopathies, and you see a prolonged QT on the ECG, as you can see there, of the 22-year-old female with an ECG with a very prolonged QT interval, shown with the red arrow. Syncope and sudden death are major complications, and this rhythm called torsade de pointe is a very bad rhythm, and which can lead to this rhythm, which is ventricular fibrillation and sudden death. I should point out at this point that there's a signature going around at the moment that looks very, very similar to ventricular fibrillation. And that might be the leader of the United States, whose signature looks very much like VF. So I don't know what it says about the upcoming election. What we've done with long QT syndrome is we've been able to define the genetic subcategories of disease. So now we can talk about LQT1 caused by a particular gene, LQT2, LQT3. Why is that important? It's important because up until recently, we treat all long QT the same. They're all in one bucket. But now we know long QT1, they're more at-risk individuals are males under 15. In long QT2, it's females over the age of 12. Long QT3, it's males, adult males. And the circumstances of the events. Long QT1, more exercise-related. Long QT2, more related to sudden noises, sudden alarm clocks, that sort of thing. Long QT3, sleep is the trigger. So we manage the patients differently depending on their subtype, and we even choose our medications differently depending on the subtype. So long QT3 is a sodium channel gene problem, and so we can now target that gene with mexilotine, which is a sodium channel modulator, which we would not normally do in the prior times, to be able to reduce the QT interval and reduce arrhythmic events. So an example where the genetic diagnosis, the subtyping of the disease, has allowed us to be more specific in what drugs we choose. The fourth area is in the area of sudden cardiac death in the young. I should have said at the beginning that all of those cardiovascular genetic disorders that I mentioned, all of them as a complication have sudden cardiac death, often young people without any warning signs. So the fourth application is the molecular autopsy, which is a fancy name for just saying genetic testing in the setting of a post-mortem of a young person who's died suddenly. So it's now mandated in Australia and New Zealand that anyone under the age of 40 who dies suddenly should at post-mortem have a blood sample collected for future use if needed. We extract the DNA, we do the appropriate genetic analysis, and hopefully find the cause of death. Why is this important? Well, in this study that we conducted a few years back, we showed that in the 1 to 35-year-old age group who have all had sudden death and have all had a comprehensive post-mortem, 40%, or basically 1 in 2 young people who die suddenly in Australia and New Zealand, No cause is found at post-mortem after comprehensive evaluation. But what we've done subsequently is take the blood samples from those cases and shown that in up to 25% of individuals, we find a genetic cause in an arrhythmia syndrome or a concealed cardiomyopathy. We've also done this in sudden explained cardiac death cases, and of recent times we've also shown the utility of molecular autopsy in SUDEP or sudden unexpected death in epilepsy syndromes in wonderful collaborative work with Professor Ingrid Scheffer and Professor Sam Berkovich. This is Richard Bagnall here celebrating. You don't often get a first and last author paper in New England, but we had an opportunity when we used to be able to not social distance to actually hug each other and celebrate together. The value of a precise genetic diagnosis in sudden death, you just have to see parents who have lost a son or a daughter. Parents ask you 1,000 questions after they've lost their son or daughter out of the blue, but the two questions every parent asks is, why did my son or daughter die suddenly, and how can we stop that ever happening again to anyone else in my family? And you can answer both those questions through genetic analysis. You establish a precise cause of death, a level of closure for the family, which is a very important thing, screening the family in sudden death prevention. So if we can target other family members who are living who carry the variant, we can initiate therapies to prevent sudden death. And the fifth area is in reproductive decisions, where we use pre-implantation genetic diagnosis, or this is offered in families where there is a genetic diagnosis known, and you can go through the IVF process and only implant embryos that do not carry the disease-causing mutation, leading to a child that is born without disease. So that obviously has substantial moral and ethical issues, but this is an area that's been sort of asked in the clinic very often once we find the genetic cause. So those are 5 very simple examples of how we're starting to translate sort of genetic information and coupled with phenotypic information in our clinics today. In the last couple of minutes, I just wanted to talk about a few other things which have actually been brought up in the previous speakers' talks, but the impact of whole genome sequencing is substantial. We're now doing much more whole genome sequencing. This was a study sponsored by New South Wales Health, where we showed in this study basically that if you did whole genome sequencing in cardiomyopathy patients, you increase the diagnostic rate by about 20% or up to 20%. Why do we increase the uptake? It's because of things like this, intronic variants, which alter splicing, which you don't pick up with panels or exome sequencing, mitochondrial genome changes, which are important, and new genes as well. So this has been great to show that whole genome sequencing is a value add in terms of screening these families for the particular mutations causing disease. And as Warwick alluded to, we're part of a study, a 600 family study of whole genome sequencing as part of Australian Genomics, which Catherine North has led and pioneered all the way through. And it's a privilege for us to be part of that study. And the final thing I'll talk about is complex genetics. Everything I've talked about so far is a single gene problem leads to disease, autosomal dominant or Mendelian inheritance, but some things are complex. So we can see from this photo that the groom's family are much, much taller than the bride's family. But to my knowledge, there's no one gene that leads to height. And if there was one, I probably wouldn't have it. But what we do know about height genetics is that it's a multitude of hundreds and if not thousands of variants which contribute to height as an inherited risk. So the whole idea of oligogenic and polygenic disease is important, and we're finding this more and more in several of the conditions that we study. We're also using polygenic risk scores, or starting to think about polygenic risk scores in more common areas of coronary artery disease in cardiovascular medicine. So this is genetics coming to the masses. Up until now, I've been talking about rarer conditions. This is now genetics for the coronary artery disease patients as an individualized risk factor for coronary artery disease. So this is on the horizon as well. Of course, I can't not show one slide of the possibility of using genome editing to try and correct these abnormalities. Only in the last week, Jennifer Doudna and Michel Carpentier won the Nobel Prize in Chemistry for genome editing technologies and CRISPR. And such a wonderful technology. We can do it in our simple little lab in the Centenary Institute. Take blood from the patient, make the cardiomyocyte cells. Here is the mutation in the line, which is the most common hypertrophic cardiomyopathy mutation. We apply CRISPR technologies and we actually get rid of the mutation. So we cure the disease in a dish, but the next step, of course, is to understand mechanisms of disease, use these models for new therapeutics and hopefully one day be able to treat humans with these sort of approaches and technologies. So in closing, I'd just like to say that precision medicine in cardiovascular medicine is really coming to the fore. It's a loose definition, but I think we've been using it more and more in targeting therapies to the right patient at the right time based on their genomic background. And there's a whole list of conditions which I haven't even touched on. I talked about long QT syndrome. I completely ignored familial hypercholesterolemia, which now has the PCSK9 inhibitors, which target the particular mutation for disease. In thoracic aortic aneurysms, genetic information helps us decide when to go to theatre in terms of operation. And then we've talked about pharmacogenomics as well. So my take-home messages, and this is my last slide, there seems to be an emerging role of precision medicine in clinical cardiology care. The knowledge of the genetic underpinnings of cardiac diseases is leading to more precise diagnosis, therapy, and I haven't touched on prognosis today, but some information there as well. The molecular autopsy can define the cause of sudden death in the young with all the benefits I outlined. Defining a precise genetic cause can facilitate reproductive decisions. And in my opinion anyway, this is best translated using a multidisciplinary approach where it's not just one person delivering the care, but a team of people. I'd like to acknowledge my team and the wonderful national and international collaborators and the funding bodies. So thank you very much for your attention.
Chris, before we move on, there's a general point which has been raised, which around genomic medicine, which relates more in one sense to Marcel's talk, but also has this implication of whether what you've been describing is applicable only to the Caucasian part of the population. So one of our members made the observation that although understanding differences in the genetic makeup of different populations is important, many of these genes have effects across multiple populations. And the particular example in Aboriginal and Torres Strait Islanders is that many of the mutations that you've described, if present in those populations, will biologically have the same impact. The questioner made the point that they'd seen an Aboriginal child with the commonest pathogenic variant in a particular condition, which is present across all ethnic groups. So just balancing this, would you like to comment on the balance of clinical cardiology genetic care in the Caucasian as opposed to the Aboriginal population and other parts of the population?
Yes, so I think there's a few comments to be made there, but the main one being that we always expect there to be differences in ethnicity in terms of the clinical phenotype, as well as the potential genetic information, genetic basis. There are some variants that we find in the Caucasian population, which are also present in other Asian, Hispanic, Black, Indigenous populations, but there are others where the particular variant is more common, and it's a common variant in those other populations, but maybe a disease-causing variant in the Caucasian population. So we need to be aware of that, and every time we find a variant in any individual, we make sure we ethnicity match in terms of looking for if the variant's present in that population or not. But in terms of understanding the Indigenous population, we're very keen on doing that, and we've run a clinic for about 12 years in northern New South Wales to try and see more Indigenous population patients to see if they get the same type of disease and what their issues are as well. So we're very wary of that, and it's an understudied area for sure.
Thank you very much, Chris.